of study compilation and case study presentation. So, since 2013, the Directive 2010-63 EU is in order, and there is the inclusion of cephalopods as higher vertebrates in the Directive, along with fish and other mammals that were already included. This Directive works for the regulation on animal welfare in research and does not require for procedures to be regulated according to certain severity levels that go from mild to severe. So now there needs to be a project application prior to experimentation. So all the experimental design has to be exhibited, and this represents an opportunity for the standardization of experimental protocols. So basically, this is figure 1 from Fiorito et al. 2015, the guidelines. It's a schematic of the project application. There are 3 main areas that we have to do. There is comprehensive justification, where we talk about the objectives of the work, the counterbalance between harms and benefits, the availability of research facilities, and so on. Then, on the second group, there's the procedures and their severity classification. And here is what I think we can, by expliciting the procedures, we could work for standardization. And the third part is the application of the 3 R's principles that I'm going to talk about just shortly because it has been mentioned on these days extensively. So the first R is replacement, so avoiding using animals in experimentation. They are replaced for other models. The authors in this paper did not replace cephalopods, but they replaced the prey used, that was crab. So they are not including the directive, but it is the start, and it is kind of the concept behind this, the replacement. Afterwards, refinement. So we have to update our husbandry conditions and the welfare. And afterwards, also design experiments that had humane endpoints and did not require the sacrificing of animals. Here in this paper, for example, the authors, instead of dissecting the animal and measuring brain size and severity relative biomass, they used ultrasound waves to calculate that. And the third R is reduction, that is to reduce the number of animals used in the experimental design. Here I chose a paper of statistics research because I think it's a very unexplored area in this topic. I think with better statistics we can also try to work to reduce our N and not just limit ourselves to the parametric, non-parametric tests, but also go deeper in it and try to work on animal reduction that way. So another source for possible standardization are the arrived guidelines that were published in PLOS Biology in 2010, I believe. They are not meant to cut down the creativity of research, but they are meant to guide authors in presenting their essential information in the paper. They are meant also to promote the reproducible and transparent, accurate experimental procedures that can also be tested in other populations and other maybe unseen species, but to evaluate interspecific and interspecific variation. And also, and more importantly, and I think this was very well made, is the checklist for manuscript preparation that I'm going to present now. And I want to give focus more on the methodology section because they clearly point out each point that should be present in a up to the regulations nowadays manuscript. So we have the ethical statement, the study design, experimental animals, the sample size, husbandry and housing conditions. Also they talk about randomization and bias checking and measures that are employed to do blind group studies. That are also very important so there are no experimental biases. So summing up, standardization, what would it allow in research? It will allow for unanimous welfare enhancement tools, for example, using cognitive tests to establish the psychological state, as was mentioned here earlier. Increase experimental reproducibility, that is also important. Sometimes there are divergences created in science that maybe could be solved by better experimental designs and procedures. Reduced experimental outcome variation, because everyone that works with cephalopods and their behavior knows that interspecific variation is a very common thing. And so it happens due to sex, life history, maturation stage, etc., etc. So we should also work in reducing that as much as we can. Lastly, I believe that this should be a development of consensual protocol papers. There shouldn't be any unilateral perspectives just presented. It should be a joint effort through multi-collaborations and multi-institutions, and to provide some cases for how standardization has helped throughout the years in research. I'm going to present 3 case studies. See, the first case study is the widely used Prawn-in-the-ER test for Sepia officinalis. Since Messenger 73, this test has been used in by many groups, many research groups, and it basically assesses the response of the cuttlefish to a shrimp inside the tube through the 3 main phases: there are tension, positioning, and seizure. Seizure with a tentacular strike. Posterior publications have hypothesized that this could be due to habituation, due to pain on tentacular strike, and the lessening of the response, or it could be due to associative learning, as the cuttlefish learns that the glass tube is there, and it starts to diminish the response. By presenting a small change in the methodology, by substituting shrimp for a crab in the tube, as the cuttlefish uses a different strategy to catch the crab, it uses the arms. It is a less painful method, and the authors argue that due to that, it cannot be habituation due to the pain, but it is due to the associative learning through multisensorial memory acquisition that learns that the glass tube is there, so it doesn't attack it because it knows it can't reach it. So this is a case that a small, maybe not that small, but a change in the experimental procedure has led to a different result and to the clarification of cryptic scientific questions. The second case is a case for reversal learning in Octopus vulgaris. So the basis of reversal learning is discriminative learning, where you use positive stimulus that is rewarded with food or play, or both, and then neutral stimulus that is not rewarded at all. So the octopus should react to this stimulus and not react to this one. I have here 2 videos. This one is a positive stimulus. These videos were made during the training school at Officinalis in Barcelona. So I have to thank everyone there. So this was the response. This one you don't see quite well, but the octopus is there behind the arm, and you only see a slight arm extension. The octopus doesn't go to the stimulus because it was preconditioned. So this, afterwards, the reversal is made by exchanging the meaning of the stimulus, and in optimal conditions it should only require 1 stimulus trial error. This type of methodology was extensively used in the 60s and 50s with some mixed results. And today, a very recent paper has found another way to present this case of behavioral plasticity, where they have removed the experimenter bias by presenting a computer-generated stimulus. So he would in the end, in the left part, for example, is a positive stimulus. The negative stimulus is in the right part. They show the stimulus, and if the octopus goes to the right panel, it is rewarded through the that F-tube there with food, and afterwards with the vertex rod to play with it. They found very consistent results, not very consistent, but more consistent results in this study, and even one octopus that was able to do reversal learning after just one trial. So there was a significant improvement in methodology by subtracting the experimenter bias. Lastly, I think that as I'm talking here about standardization, it is also good to inform the community of something that maybe not everyone is aware of. So this case study is based on a study that was aimed at removing hemolymph and removing hemocytes from the bobtail squid, from the Hawaiian bobtail squid, to then test their adherence with symbiotic and non-symbiotic bacteria. It was published in a journal that's called the Journal of Visual Experiments, that allows for a different structure of publication where you can publish a step-by-step protocol for other research teams to mimic perfectly. As you can see, they have detailed information on anesthesia and post-recovery, with ranging almost every aspect of the procedure. And not only that, they also have a video where they show all the procedure from top to bottom, and they explain the procedure throughout it. It has the same structure as the article, so I think it's a very good way to disseminate a procedure practice and to increase the reproducibility of all of the work that we do. So this is me skipping the video, so you're not here for 5 minutes. This is the anesthetic part. I believe they did with ethanol, 2%. You can see the coloration starting to go down. Then they retrieve the hemolymph from the main cephalic aorta vessel. Sometimes when writing papers, the methodology is not super clear, and this is a very good way for you to show exactly what you are doing, how you want it to be done, and to disseminate how you made your science. And then you have the stain, perfect. And this is also an application of cephalopod research in microbial research. It is out-of-the-box thing that I like. So, lastly, I want to thank everyone at Cost Action. Joanne is laughing for the photo. And everyone at the training school in Barcelona. I want to say thank you to Graziano and to Giovanna for inviting me for the presentation, for helping me out with it. Also, Viola Galiccioni, that she's not here already, and Andrea Taral for helping me with the abstract. And I would like to say thanks to this crazy guy that is my supervisor, that makes me believe I can do 5, 6 things at the same time. And then we stay awake until 4 o'clock and miss the visit to Knox. So thank you all.